So welcome all to this conference. I should start by saying that it was meant to be a small scale affair, but I'm really happy to note the kind of enthusiasm and participation we have seen. We have actually participants coming from as far as IIT Jammu for this conference. They have made their own arrangements. I thank all the participants, especially who have come from outside Chennai and making their arrangements. And this was meant to be a conference where we have speakers who are largely open-minded. I don't think there was any specific theme, but most of the speakers that you will see here in this conference have a quantitative bend. Some, in particular, majority of them do only computational work, but some do a mix of both computational and experimental work. So that's essentially the broad theme of the conference. We are an open-minded bunch, and we are willing to discuss and be creative about various ideas. With that in mind, I hope that you will enjoy the next 1.5 days. In particular, I think some of your resolve towards science would be tested tomorrow afternoon, when I would try to see how many of you really missed the match and be here inside this auditorium. Especially some of the speakers in that session are worried. So I hope that some of you are motivated to be here tomorrow as well. So IMSC, is a small but beautiful institute. I welcome all of you here who have been from outside IMSC. So please try to enjoy the environment. We have a very open academic atmosphere where we are free to think about any research problems. So I hope that you enjoy your stay here. And with that, I'll basically welcome Sri K. Venkatrama Sharma from NCCR Chennai. He will chair the first session.
My relationship with Professor Samal is recent, but I have already seen how determined and dedicated he is. He's going through a period of personal stress, but to put on this show is a humongous achievement. So my compliments to you, sir. Today, in this opening session, we will have 3 speakers who will talk about how they use diverse biological data, computational techniques, and mathematical algorithms to make sense of how genes do their stuff. So without any further ado, I would like to request the first speaker, Dr. Rahul Siddharthan, to come onto the stage, please.
Thanks very much, Dr. Sharma. And thanks, Arijit, for organizing this meeting. And I guess thanks for putting me as the first speaker. So Arijit has already introduced the institute and the Combio group, but he asked me to say a few words about us in any case. So we started this computational biology PhD program in 2013. So it is, we took our first batch that year. So it's basically exactly 10 years now that we've been running a PhD program in computational biology. We started as a bunch of people from the physics group who were interested in modeling biological systems in various ways. And I think the program has been a reasonable success. We've graduated quite a few PhDs by this time, some of whose work I will talk about now. And we kind of been working with a lot of the best biologists in India and abroad for the last 10 years or so, and we get quite a lot of interest. So for those of you who might be master's level students who are looking for PhD programs in computational biology, or if you know anybody who are, our applications are open every year. And also if you know anyone who's looking for faculty positions, we are always welcoming of people who would like. But even otherwise, anyone who wants to work with us, to collaborate with us, just look at our webpage. Our individual web pages, our group webpage. So with that, I'll talk about, I'll switch to my topic. So the conference is on modeling and tackling complex biological systems. And I don't really do complex systems in the physics sense of the word, but of course, biology is a complex system, and I'm talking about transcriptional regulation. I'm just going to talk about 2, and if time permits, 3 pieces of work that we've been doing over the last few years, but just as a broad overview. But first, let's just reintroduce what DNA and chromatin actually looks like inside a cell. So as most of you would know in this audience, eukaryotes have a tightly packaged DNA and protein complex inside the nuclei. And this has a hierarchical structure which starts with double-stranded DNA, which the canonical Watson-Crick structure, but that in turn is wrapped up around these histone complexes, which form octamers, and they are sort of disc-like protein complexes around which the DNA binds. And these green things hanging out are histone tails, which play an important role in all kinds of regulatory processes. And so this complex is called the beads on a string model, where you basically have roughly 150 something base pairs of DNA wrapped around one nucleosome on average, including a 30 base pair linker roughly. But these guys are not static. They can slide around, they can be popped in, popped out. And then this is only the sort of secondary level of the structure. Beyond this, you have this assembling into a sort of helical structure, which is called the 30 nanometer chromatin fiber. And that in turn is packaged and into other layers into chromosomes. And your entire DNA is divided in humans into 23 pairs of chromosomes. And in other species, it varies. But this is basically how chromatin looks. Now, why is this important? Well, so what I'm talking about is transcriptional regulation. That is how genes are turned on and off. And so within DNA, there are certain regions that code for, they're called genes. They either code for a protein or they code for an RNA molecule. But in either case, you need to make an RNA molecule, which is either a messenger RNA, which gets translated into a protein, or it's the end product in itself. And these genes, of course, need to produce their product only when they need to. And that's why different cells look different in your body. So this is just a cartoon from Wikipedia, but it kind of illustrates why the packaging is important. So the gene gets turned on by various regulatory proteins, which bind either near the gene in a region called the promoter, or maybe very far away from the gene in regions called enhancers. And these are far away in sequence space. So as computational biologists, we think of DNA as a string of ACGTs, which is a few 100 million base pairs long for each chromosome. But in fact, DNA is not a string, at least it's not stretched out in a straight line inside your nucleus. It is wrapped around in complicated ways, as we saw in the previous slide. And so in this particular case, these enhancers are brought in proximity to this gene, and proteins that bind here might turn on this gene. And these proteins, which are these pink circles, are required to basically recruit the RNA polymerase, which makes the gene RNA product. It transcribes the gene into an RNA molecule. And another layer of regulation goes at the translational level, which is when the mRNA needs to be translated into a protein. So we won't go into that here, but many of you know about that RNA and siRNA and so on. And so that we won't go into. But how is this process regulated? So first of all, even the action of an enhancer can be modified by certain regions called insulators. And there's a factor called CTCF, which we'll talk about quite a lot, which was originally identified as an insulator binding factor. It stands for CC-CTC binding factor because the binding sequence that it prefers tends to be C-rich, though it's a bit more complicated than that. So when CTCF binds to an insulator, it has the effect of modifying the chromatin structure such that the enhancer no longer works. And then you also have chemical modifications to the DNA, such as methylation. So if you have, if you turn the C in DNA to methyl C, that has an effect on its function in terms of being able to bind proteins or in terms of transcription and so on. So basically, it's an extremely complicated system. So the two things we'll talk, okay, and how do we probe all this experimentally? These days you have a whole array of next-gen sequencing based techniques. I talked about transcription, And so these techniques talk about how to measure the levels of transcripts of RNA molecules in a seq. So RNA-seq is kind of the most common technique. In the previous era, you use things like microarrays, which are still used for some purposes. Then I talked about methylation of cytosines, and that's done by something called bisulfite sequencing. Then binding of specific proteins to DNA, and also assaying for certain chemical modifications to the histones. Both of those can be done via a technique called ChIP-seq, which we'll talk about briefly. And for identifying which regions of chromatin are relatively open, that is relatively free of nucleosomes, is done via these techniques, DNA-seq, ATAC-seq, and so on. And finally, how the chromatin is structured can be proved via this kind of technique, which tells you in various ways which regions of the DNA might be in contact with each other, right? So this is an array of things that experimentalists do, and projects like ENCODE have databases across hundreds of cell lines and, transcription factors and so on, various combinations, but very far from every possible combination. Okay, so how is chromatin organized? I mentioned CTCF. This is from the Murney lab, and there's a YouTube video called the loop extrusion waltz, which I'm not sure that I can play it here, but I have some slides. But essentially, you have something called, you have something called a cohesin complex, which forms a ring-like structure, and this black thread is supposed to be the DNA. And the DNA gets loaded onto the cohesin gets loaded onto the DNA, and it kind of gets pushed through. That's the picture they have. And this was a sort of biophysical picture that I think they kind of came up with even before a lot of it was experimentally confirmed. And these red things are CTCF binding sites, and they act as barriers to this extrusion process if they are bound and oriented correctly. In this case, both of these CTCFs are oriented in a parallel way. This one will act as a barrier, and this one won't, so it goes right through. But then this guy is coming along, and this does act as a barrier because it's oriented oppositely, and that halts the loop extrusion. So you sometimes have one-dimensional extrusion, your bi-dimensional extrusion. And I should say that a lot of this is specific to vertebrates and so on. So if you look at yeast, which is also a eukaryote, it doesn't have CTCF, but it has other proteins that model chromatin. So CTCF, I mentioned, is an insulator. It's known to modulate chromatin structure like this. It also plays a role in transcriptional activation, repression, and so on. So it's a protein of interest that we are trying to bioinformatically study for a while now. How do high-C experiments work? Basically, the idea is, so all these sequencing experiments basically break up DNA into lots of small sequence fragments and then run them through a sequencer. And in the case of ChIP-seq, you isolate the fragments that were bound to a protein of interest, which you pull down by an antibody, and you map the reads to the genome to see where the peaks come. In high-C experiments, the idea is that you cross-link the DNA, so pieces of DNA that are kind of in contact get cross-linked like this, and then you do a restriction digest, and then you ligate them, so that what you have here is a chimeric DNA where you have two pieces of DNA that were originally from two different parts of the genome are now on the same sequence fragment after you reverse the cross-link. And then depending on which of these protocols you're using, you might be looking at specific regions like 3Cs local, 4Cs like one versus all. So this is like high-C is the most common. It's all versus all, genome-wide you're doing this assay. We'll also talk about ChIA-PET, where you are tagging one of the ends to a protein of interest. So you're looking at which parts of the region things that protein binds to could interact with. For example, the protein could be Pol2, which is an RNA polymerase subunit. So that might tell you which enhancers interact with which promoters, for instance. We'll come to that. But basically, when you sequence these chimeric reads, you see where they map to the genome, and those two places they map to are the ones that are likely to be in contact. And the sort of output from these experiments looks like these heat maps. And they basically, the intensity of the red tells you, or in this case, the intensity of the red or orange, tells you how probable contacts between these are. And contacts among the diagonal are, of course, the most probable, but you see significant off-diagonal contacts here and there. So if you zoom in, this is what it looks like. And you also see these sort of triangles on the diagonal, and these are signs of these topologically associated domains that we talked about. Okay, now with that, we'll come to some of the things that we've been working on. So, well, before that, I talked about ChIP-seq. Let me just review what ChIP-seq is. You are interested in where a particular protein binds. And so you have an antibody specific to that protein, and you send it in, and you cross-link the DNA protein complexes, and you sonicate it. And well, you do that first, and then you send the antibody, and it pulls down the fragments of DNA that your protein is bound to, and then you sequence those. But when you do the experiment, you often, and then you do a motif analysis on your ChIP-seq peaks, you have a protein that you know binds to a certain motif. And you often don't find that motif, you find a whole bunch of other motifs. And these people talked about this in 2014, and they called them zingers. In particular, certain factors like ETS and CTCF itself and others, their motifs tend to crop up across many different ChIP-seq experiments. And this is a kind of cartoon of their example of why this might happen, that you might have in your ChIP-seq experiment, you might actually have a direct interaction between the pink protein, which is your zinger, and the blue one, which is your ChIP-seq thing. You might have some kind of an, so here they are actually bound to the same neighboring region. Here they are bound to a region which is in 3D proximity. It's not really neighboring, but if you chip for this, you might still pull this down because of this cohesin ring. And there are various other things like cofactor forming and non-specific binding that could complicate the issue, but certain factors seem to show up in these data. Okay, so one of the first things we did in the lines of analyzing ChIP-seq data was this, and I'll just quickly overview this. Ankit was I think our first PhD student to graduate, and he worked mainly with Gautam Menon, but he did this piece of work with me. And He's an old collaborator whose name will come up again. Snehal was a project assistant. But essentially what we did here was we took ChIP-seq reads, and instead of running a motif finder, we tried to cluster them in terms of sequence similarity. And for that we developed a sort of an algorithm that does this quite efficiently for large numbers of sequences. And if you do that and then you try to plot the clusters using, visualize them using sequence logos, which is what you would do for motifs, we in fact find that for any data set we looked at, like for IRF1 or for REST or NFI or FOXA, we do find the canonical motif jumping out, sometimes with additional substructures, but we also see other motifs coming out, like ETS I mentioned earlier, it crops up a lot. Something which looks like SP1 crops up, something that looks like these two are CTCF, which crops up. And the other thing about our algorithm was that we were able to pull out examples like this, where this is the canonical REST motif, it's a heterodimer. This is the same motif but with a bigger spacing, and most motif finders have trouble finding this. This was actually known in the literature, but people found it by looking for the two halves separately and putting them together. But we were able to pull this out of standard data sets. But again, we find variants where this is just like 1.5 copies of this, and this is like 1 copy of this, and this is a CTCF motif which crops in. So there's a lot of stuff like this. And in terms of zingers, these are, in addition to the ones that Kant and Wasserman reported, we find these motifs occur across a bunch of different ChIP data sets. So IRF1 occurs in all of these, SP2 occurs in all of these, and many more. And again, the REST motif occurs in both versions in data sets which are not ChIP for REST at all. Right, so this was interesting, and we are, so some of the conclusions from this work was that these Zinger motifs are common, but also we found additional structure to a lot of motifs. Like here we found an extra piece over here. In CTCF, we often find additional motifs here. We find an extra motif here. So this kind of thing is very common. So the picture traditionally is that a transcription factor binds to a specific region and it prefers energetically a specific sequence there. But what we find is that in a lot of flanking sequence, you have some sequence signals. And increasing many people are observing the same thing. Okay, so with that, the sort of main work I wanted to talk about here is this one, which got published earlier this year. Rakesh was a very bright student here who's now in the Dimple Notali lab at NCBS. He specifically wanted to work in an experimental group after this experience, so I hope he does well. So here, what we did was look at, so in transcriptional regulation, it's not just one factor regulating a gene, it is multiple factors regulate a gene. And these factors can all bind on the same promoter, or some could be on a promoter, some could be on an enhancer. So as I showed you several slides ago, enhancers or promoters are brought in close proximity by 3D structure. So can we ask which transcription factors like to interact with which other transcription factors? And thanks to the large amount of Hi-C data available and ChIP-seq data on the same cell lines, we could kind of answer that question. What we did was, in 3 dimensions, can we determine whether two pairs of two transcription factors, let's say TF1 and TF2 here, occur more or less often than chance in 3 dimensions? So by co-occurring, we mean that they are both bound to regions that are known to interact. So this network is a network of genomic regions that interact according to Hi-C experiments. They are not spatially contiguous, but the contact map shows that they have interaction. And this is the binding network of TFs to the same network. What we do is we randomize this binding network, keeping essential properties the same. In particular, the coordination number of each TF is the same. The coordination number of each binding region is kind of the same. And so if one region binds to many TFs, we'll let it bind to many TFs. If one TF binds to many regions, we'll let it bind to many regions. A bunch of such constraints that respect the essential network topology. And then we can ask for something like 60 TFs in GM12878 cell line, where we have ChIP-seq data and we have Hi-C data. Which ones tend to occur? So this is a heat map where you can get a Q value from this randomization. You can get a P value by saying, it's basically the fraction of times that they co-occur in random networks. And you can convert that to a Q value. You could also ask, do they not occur together by chance? And a Q value is basically false discovery rate. I won't go into that, but these show the Q values. And the greens are significant Q values of co-occurring, and the reds are significant Q values of not co-occurring, and black means not significant one way or the other. And what we find if we bicluster those greens and reds is that they fall into 2 very nice blocks of genes. One block is here, they talk to each other, and one block is here, they talk to each other, and these 2 blocks don't talk to each other very much. There are a few exceptions, like the central block of genes. If you look closely, you'll find that it consists of SMC3 and RAT21, which are both coezine subunits. It has CTCF, which also is involved in chromatin structure, and it has YY1. So these genes seem to be like friends with both blocks. They can talk to both blocks in a sense, but they talk to each other more. Actually, yeah, it's a bit more complicated than that. In other cell lines like K562, we don't find such a clean breakup, and this is a different set of TFs, though overlapping, because we don't have the same ChIP-seq experiments in all cell lines. But here again, we find one core group of genes that tends to interact with itself and not so much with other genes. And in fact, this was actually observed in GM12878 using a different methodology by these people. And so despite being kind of scooped very early on in our work, Rakesh pushed on to find a lot of more interesting things that could come from this kind of analysis. So one point is that this biclustering is reasonably robust. The essential features, in particular this green block here, are conserved across multiple cell lines. Then we looked at what about if two transcription factors tend to co-occur in 3D, according to Heisey, do they also tend to co-occur within the same sequential region, within a promoter, for example? And it turns out if you take the same set of TFs, but you ask whether the binding sites tend to co-occur within a region more often or less often than chance, you get very similar heat maps, but kind of stronger. There are fewer black regions. This is a sort of significance plot that I won't get into, except brighter blue means more agreement. So we looked at a bunch of other things like, so how do these two groups of factors differ? We looked at the HIPPY database for protein-protein interactions. When I say we, I mean Rakesh really. The two groups sort of differ in, so this is the first group one is the green is this group, and group two is this group. And the set of proteins that occur in both sets of interactions are shown in brown, and there is a small set that look like that. These are central nodes. So the idea is that within the network of interactions that connects two pairs of proteins, which are the proteins that crop up more often, and so which possibly mediate these interactions. So some of these, like YY1 I mentioned earlier, is the chromatin factor, and some of them might be kind of expected, but most of the interactions are actually different. And if you take pairs of genes from one group and the other, and ask what are the central nodes there, then you see this kind of a plot. And again, these brown guys, most of them co-occur here, but the rest are a mixture from the two groups. Is there any, okay, this is kind of important that the group one tends to be much closer to a transcriptional start site on average than to the nearest TSS in the genome on average than group two, which means group one is likely more associated with promoters, and group two is more likely associated with enhancers. Looking at the gene function, I'm sorry if this is not really readable, but group one is sort of more enriched in housekeeping genes, which, so some of the things written here are like heterocycle biosynthetic process, RNA biosynthetic process, and so on. And group two, it depends on which cell line you look at, but some of them in GM12878 were like related to immune response and other kinds of signaling things. So it looked like group one tends to be the highly conserved group in terms of evolutionary function. Their housekeeping genes, they're really important. They sort of work together. Group two are perhaps the ones that are more disruptive. Also to be mentioned, like K562 is a cancer cell line. So genes that in group two in that might be linked to disrupted function in a sense. The last thing was, and this arose from previous work from Sridhar, supposing you have two factors in 3D space that are kind of close to, in 3D their binding sites are close. Does the presence of a factor here influence the probability of binding of another factor there? In other words, do factors actually cooperate when binding? And if so, how can you tell? And one way you could tell is that if you're getting help from this other factor, you can get away with a weaker motif. Whereas if you're getting no help, you will need a stronger motif. And Sridhar had actually done this for homotypic factors in an unpublished work that is still on bioRxiv, I think. But we wanted to look at it for heterotypic factors. That is, if you have factor A and factor B, or actually you don't care really what there are any two factors. If you have a binding event in one region, does it influence binding in another region? And it turns out that it does show up in a difference in motif intensity. If you have a spatially interacting ChIP-seq peak, typically you have a weaker motif. This is the motif log odd score for the binding sequence plotted as a thing. And in almost all cases, there are some exceptions, of course, but in most cases, the yellow dots are below the blue dots. It's quite a noticeable effect. So this is so these are the conclusions of this piece of work that the TF-TF associations form two distinct groups which are mirrored in sequential interactions. Groups differ in proximity to TSS in PPI in various functions, and one group is housekeeping and the other possibly regulates lineage-specific functions, and these things could be disrupted and considered as kind of speculative. And this complex pattern would have co-evolved with the genome, and you can. So we provide an open source tool that people could use if they want for studying other data sets, other cell lines, and I think this kind of work will probably get pushed in the future by various groups. Right. So in the last 7-8 minutes, I'll talk about some much more recent work very briefly by Pavitra, who's I think in the audience here. She's also finishing soon. And this is about how binding sites evolve. And I have way more slides than I can present for this, so I'll just give you the big picture here. So if you looked at those motifs, you can actually, you have genomic sequence for a whole bunch of mammals these days, including a whole bunch of primates, and you can map a human binding site to sequences in chimpanzee, mouse, whatever, and ask how similar are they. What we wanted to ask is what are the selective pressures within the binding site, and there are various models of DNA evolution that have been published since the 70s that deal with exactly this problem. Typically, so the first thing is, how does DNA evolve? Okay, I'll skip this. DNA evolves via these processes, single nucleotide substitution, which is all we'll talk about here. Because we are talking about transcription factor binding sites, insertion deletion is likely destroy the function of that site because you need a motif to be intact. And all these other things also affect how, so you might have multiple copies of a gene where a mouse has fewer copies. You might have a different order of genes. All these things affect your evolution, but we are only talking about the first point here. And there are models of single nucleotide evolution which have been used forever, basically. But here's an example. You might have, this is completely made up, but you have 5 sequences from 5 species. You can look at their distance metric between them. So just count of the mismatches of nucleotides gives you a possible metric, but you want something more closer to evolutionary time, then you need an evolutionary model that tells you how many mutations you expect per unit time, and so that you can map this conserved fraction, because you will, even if 2 species are completely diverged, you expect 25% conservation just by chance. So the Jukes counter model is the simplest thing that does that, and there are more sophisticated models. And that might give you, then with the distance matrix, you can construct a tree that might look like this. So I think I'll skip past the math here, but essentially what I will say is you end up with a tree that looks like this, and there are 2 parameters to the tree. One is the topology of the tree, which for our purposes we take from the literature because we know human is closer to chimp and mouse is further away and so on. And the other is the branch lengths. So these Qs in our notation are some version of the evolutionary time. It is really the conservation rate, so it's e to the minus mutation rate times time. But never mind that. Given a tree like this, you can calculate the likelihood of the leaves that you see on the tree. So supposing this is human, this is chimp, and this is mouse, and this is A, this is C, this is T. How likely are you to see these 3 nucleotides? You can calculate that exactly using this tree and using the transition probabilities that your mutation model gives you. And since alpha and beta are unknown ancestral nucleotides, you sum over them. So basically, this work, I'll skip this. One of the things we have is that we can actually do this very fast for a particular model called the F81 model, which for a tree that's not too big. I'll skip that also. But the big picture is that if you go back here, what we are going to do is estimate the branch lengths using neutral evolving sites, which other people have done, but we do it just for put things in our own picture. And then any evolutionary model gives you an equilibrium frequency at each locus. And that means if you let it run forever, what is the distribution of nucleotides? So A will mutate to G, to C, whatever. But if you have a population which is evolving, what is the distribution of A's here? What is the distribution of C's here? And so on. So that is called the stationary distribution. And it is position specific. That's what we assume. And so what we ask is, can we retrieve the position specific distributions of nucleotides within the binding site from this kind of an evolutionary model? So that was the question. And yeah, we have a fast way to do it. I'm skipping that. Also, can we infer the most likely ancestral nucleotides at any position, which we don't directly need, but I'll come to why we. So if we can do this, then we can actually condition on the ancestor and ask, if the ancestor was A, then what are the pressures? If the ancestor was C, what are the pressures? So with that, I'll skip to the results here. So our old friend CTCF looks like this. We've been interested in CTCF for a while because of these complicated dependencies that you might see. So for example, if you look at Drosophila CTCF, this site typically is a G, but in vertebrates it's often an A. But when it is an A, if you look at, you won't see this in the logo because this logo assumes everything is independent. But if it is an A, you often see a G next to it. Whereas if it is a G, you often see a C next to it. So if you condition the ancestor to be an A, or if you condition it to be a G, do you see a difference? And in fact, you do see that if the ancestor is a G, you see a difference here. But you also see differences in a lot of other neighboring nucleotides. So like this C becomes less important. And so the heights here are basically Jensen-Shannon distances between these two frequencies. There are other positions that are also important, like this one seems to be relatively important for the next guy. This one, on the other hand, it's a similar A-G polymorphism, but it doesn't seem to have much effect on neighboring. It doesn't matter what the ancestor was. The rest of the logo looks pretty much the same. One question that a reviewer asked is, can you do a control with, a randomized PWM? So we tried scrambling the CTCF motif, and we find that the effect is much less. First of all, the PSSV, this is the selective pressures. Here it is almost as strong as in CTCF, but here it is much weaker, which is understandable because these scrambled motifs don't have any functional role. We see similar results with a lot of other factors. Two of them are shown here. So for example, for rest, which I talked about in the earlier work, this particular position is either C or T. But it looks like if you have a C here, then this first half of the rest motif is much less important, and the second half is more important. If you have a T here, then both halves are important. So this, the choice of the ancestral nucleotide here has this kind of effect on selective pressures that goes through the rest of the thing. To a lesser extent, you can see that here also, whether this is G or A affects the rest of the motif. There are many other results that, and we have notebooks that we can use to work out. This is basically, we used a model called F81, which some might say is oversimplified, but we used it for our purpose. We think it's actually correct. But there is a slightly more complicated model called HKY85, where we cannot do the fast calculation I talked about, but you can do a reasonably fast calculation. It takes hours instead of minutes. We get basically similar results with this model, and that was the purpose of this slide. Okay, so that was this work, which was just out a couple months ago on bioRxiv, and hopefully it will go through sometime soon. Yeah, so these were the three things that I thought I'll talk about today. It's just a sort of broad overview of how complicated this field is and some of the things that interest me personally. So thank you for your time. Is there time for questions? Or?
Nice work, Rahul. So for the last part, are you also taking the protein evolution also into account to see if the both sides of the coin have come together to this evolution?
Yes, I think in this case, most of the work was primates only. We don't, even within the mammals that we see, we don't expect the protein to evolve in this time scale in terms of its binding affinity. So CTCF or whatever these factors are, they have pretty much the same binding affinity across these species. If we were to look at something like yeast, then the protein also will be very different, or not even fruit fly or insects, but those are so distant that reliably finding homologous binding sites may not be, the question may not make sense. Because what we're doing is we're aligning binding sites from human to corresponding binding sites in chimp, rhesus, whatever, which you can do reliably within primates and to a lesser extent within mammals. So I think protein evolution probably does not play a role here, is my feeling.
Okay, cool. Thanks.
Thanks for the talk, sir. Great talk. I had a question regarding the binding network. I wanted, could you please tell more about how you randomize the edges?
Right.
If there was an algorithm for that.
Yes. Like I said, I think Rakesh did this work. But essentially, you make a network. This is a sort of the cartoon that we came up to present it. So the numbers here are the coordination numbers. These are not. There's just example numbers. Supposing you had a real world network like this, and this is derived from Hi-C data. So, and A, B, C, D, F, G, H, I are different regions in the Hi-C data. Then according to this, region C has 3 interacting partners, so we've put that here. And what we're doing is so if we take a link from TF1 that goes to a region with coordination number 1 in this network, we reassign it maybe to the same node. So we could keep it here. But okay, let's take the link that goes from B, which has a coordination number 4. We reassign it to node G, which also has a coordination number 4. So we preserve that. We also preserve the coordination number of each TF within this network. Like if TF has 4 links, we retain 4 links. But there is some flexibility here. So here in this cartoon, it's exact, but we actually have constraints like within less than 5, we maintain it exactly. If it is 5 to 10, we keep it as 5 to 10, and if it's more than 10, we keep it as more than 10. That kind of mapping we did.
Thank you.
Thanks.
Here.
Hi, Dheeraj.
Nice talk. So I'm just curious about the TF-TF association, the metrics that you showed.
Yeah.
So I assume, your list of TFs on vertical, horizontal axis are same, right?
Yes.
So why is it not following the standard diagonal pattern, and how you could?
No, it's symmetric. These are symmetric. The diagonal is this way.
Right, but I would assume that in the same TFs should show the co-association. Why that is not visible here?
Oh, they don't all. Most of them do, but they don't all do. And but none of them. See, the black means that there is no significant association.
No, I'm following the heat color map towards the right.
This one also. Sorry.
No, the heat color map at the extreme right, where you show the.
So the green means associate, red means avoid. So basically, this is 1 minus the. So we calculated Q values for 2 cases that don't associate and that do associate. And both cases, the Q value is like less than 0.05. We plot it if it's more than 0.05. But for the ones that don't associate, we are just plotting 1 minus Q.
Yeah, just curious that how you would get don't associate for same transcription factor.
We don't get that. So I don't think we do get in a few cases here. And right, I don't have an answer to that. But for the most part, even within this block, either it's black, which means it's not significant, or they do co-associate significantly. Not significant means, it doesn't care whether it's, so there are factors, I guess one should look at the individual factors to answer that question. So that's an interesting question that I hadn't thought about, and it doesn't occur in this data. So it could be something to do with the K562 cell lines nature also, I don't know. These cancer cell lines are kind of, yeah.
Thanks.
Hello, sir. Sir, I'm interested to know where the cohesin protein molecule extrudes on the chromatin fiber towards the CTCF protein molecule. So what is the molecular mechanism used here that pushes the cohesin protein towards the CTC effector? Or is just a random diffusion?
See, the cohesin protein is not being pushed towards the CTC effector. It is just that it is extruding a loop until it hits the CTCF that is appropriately oriented. It's not, this is the point where it stops. But what is causing the extrusion, what's the energetics, dynamics of that I'm not an expert. Some other people in the audience might know.
Okay, sir, thank you.
Yeah, thank you.
So here, I would like to know what's the application of using the ancestral nucleotide sequence that you had constructed. Is it like to tweak the binding specificity of the transcription factor, or what is it? Because at the protein level, generally they try to design sequences using ancestral protein reconstruction and try to design some enhanced function or something. So what was the objective? Maybe I missed it.
The objective here was just to see whether there are, just to backtrack, maybe this part of the talk was too fast. A position weight matrix assumes an independent distribution of nucleotides across positions. But in fact, we know that they are not independent. There are significant dependencies. For example, I pointed out that G tends to be associated with C. And so what we wanted to see was, if ancestrally you had an A there, do you have a different selective pressure, and can you see that? Versus if ancestrally you more likely had a G there. It's probabilistic. So how we've done this is the ancestor has a weight of more than 0.5 for A or for G. We asked for that, but if it is more than 0.5 weight, it looks like the selective pressure is actually almost entirely A or almost entirely G. But we want to know what is the effect on other parts of the sequence. And these yellowish lines are indicating the Jensen-Shannon distance in some units. It's times 5 or so. So just as a sort of visual aid. So those are the positions that the higher the yellow bar is, the more the effect is. But our question was basically, are there dependencies within motifs, and are those there evolutionarily?